This video is on the two-signal hypothesis to activate naive T cells. These signals require a T cell receptor, TCR, and co-stimulatory molecules. This two-signal hypothesis is a misnomer as it actually takes three signals to fully activate the T cell. Remember, there are two types of T cells that emerge from the thymus, the CD4+ and the CD8+ cell. These cells are called naive because they have not yet met an antigen. They circulate through the vascular and lymphatic system looking for a matching antigen. Then they become activated. When a T cell finds an MHC antigen complex that it binds to, they form a central supramolecular activating complex, or CSMAC. The affinity of the connection can be enhanced by the CD4 or CD8 co-receptors. Remember, CD4+ for MHC class II and CD8+ for MHC class I. Several other adhesion molecules circle around the TCR-MHC complex and they form the peripheral supramolecular activating complex, or PSMAC. This starts the cascade of events that leads to the T cell activation. Once the TCR-MHC antigen, CD4 or CD8, and adhesion molecules attach, tyrosine kinase called LCK is triggered. LCK lies on the inside of the T cell membrane near the tails of the TCR complex, known as TCR-CD3. The TCR uses CD3 to transduce the signal to the inside of the cell. CD3 uses 4 different anchoring proteins to do this: gamma, delta, epsilon, and zeta. The whole protein structure is anchored to the membrane as a unit. LCK phosphorylates the ITAMs that then phosphorylate ZAP70. ZAP70 activates the linker protein of activated T cells, or LAT, and its coprotein SLP76. LAT and SLP76 link to several other important proteins to form complexes of new proteins. Phospholipase C-gamma, or PLC-gamma, which breaks down phospholipid PIP2 into IP3 and diacylglycerol, or DAG. IP3 will release calcium from the ER, which connects to calcineurin and calmodulin to activate NFAT, or nuclear factor of activated T cells, which enters the nucleus for gene activation. DAG will activate protein kinase C-theta, or PKC-theta, to activate nuclear factor kappa beta, or NF-kappa beta, for gene activation. DAG also activates RAS, which triggers a MAP kinase cascade that activates AP1 in the nucleus for gene activation. Important co-stimulatory receptors are CD28 and ICOS, or inducible co-stimulatory. These molecules enhance the production of TCR and the survival of the T cell. All CD4+ T cells have these co-stimulators, and 50% of the CD8+ cells do. CD28 on the T cell interacts with CD80-86 on the APC. CD28 works through PI3 kinase to strengthen cell survival and division. ICOS plays a role in maintaining the metabolism of the T cell. The third signal is provided by cytokines produced by the APC, paracrine, and the T cell itself, autocrine. One important cytokine is IL-2, which helps in the T cell proliferation. If the TCR combines to the MHC with co-stimulation by CD28 combining with CD80-86 and other cytokines are released, the T cell is activated. If either there is no connection of CD28 with CD80-86 or there is no release of cytokines, the T cell is anergic, or unresponsive. This whole complicated process of activation is important for T cell survival, proliferation, and differentiation. This concludes the video on the two-signal hypothesis.